Imagine engineering a billion-dollar state-of-the-art cellular therapy to cure cancer, right? You drop it directly into the tumor, and it just fails because the cells essentially starve to death.
Yeah, it's incredibly frustrating, but that is the reality of CRT cell therapy today. The cells just run out of fuel.
Right, which is wild. Welcome to today's deep dive, by the way. Today we are unpacking a really fascinating 2026 paper from the journal Cell.
It's a great paper. It completely bridges everyday lifestyle factors, specifically diet and metabolism, with advanced cancer treatments.
Exactly. So the core question we're bringing to you today is, can the specific molecules generated by what you eat actually make cancer-fighting immune cells work better?
Right, because for the longest time, nutrition and synthetic immunology were treated as, totally separate worlds.
Completely separate. Okay, let's unpack this. Imagine an advanced biological super soldier, right? And you drop them into a harsh, completely starved environment.
Yeah, that's exactly what a solid tumor is. It's a nutrient wasteland. And we are going to explore how one specific molecule acts as both, like rocket fuel and a software update for these engineered cells.
I love that analogy. So let's start with the basics here. CRRT cells, these are chimeric antigen receptor T cells.
Right.
It's basically like taking a patient's natural immune soldiers, bringing them to a lab, and installing a highly specific GPS tracking system on them.
Exactly. So they can hunt down very specific targets, like CD19 on B-cell lymphomas. And in the lab, they are incredible at this.
But then they hit what researchers call the metabolic bottleneck when they go back into the patient. What exactly is the mechanism of that problem?
Well, T cells have very limited internal fuel stores. And inside a tumor microenvironment, resources like glucose are incredibly scarce.
Because tumors are defined by the Warburg effect, right? They are highly glycolytic and just devour all the local sugar.
Precisely. They are greedy. So the T cell arrives, it can't generate enough energy to keep up the fight, it gets metabolically exhausted, and the therapy just fails.
Let me push back on that for a second, though. If the problem is just a lack of fuel, why can't we just give the T cells more standard sugar? Or, engineer them with more GLUT1 transporters to grab more glucose?
It's a totally unwinnable arms race. If you flood the environment with glucose, the tumor is just going to eat that extra sugar and grow even faster.
Ah, right, because the tumor is already better at consuming it.
Exactly. You're forcing the T cell to fight on the tumor's home turf. What we really need is an entirely different fuel source.
An orthogonal fuel source.
Yeah, a fuel the T cell can use, but the tumor can't.
Okay, so let's do a quick subtopic summary for you listening. First, CRT cells are essentially GPS-guided immune cells.
Right, and they fail when they run out of energy in that harsh tumor environment.
Exactly. So here's a question for you to think about. If you were designing a super soldier cell, how would you keep it fueled in a hostile environment where the enemy literally eats all your standard rations?
And that question is exactly what led researchers to screen 6 completely different diets on mice.
Right. They used an A20B cell lymphoma mouse model, and they wanted to see which diet helped the CRT cells the most.
And out of all 6, the clear winner was the ketogenic diet, or KD, a high-fat, ultra-low-carb diet.
But here's the reality check. Placing a really sick cancer patient on a super strict keto diet is... it's incredibly hard, right?
Oh, it's practically impossible for many of them. They often already have severe appetite loss and gastrointestinal issues.
Right. So the researchers were like, okay, we can't do the whole diet. What is the active metabolic byproduct of this diet?
Exactly. They isolated the primary ketone body produced during the diet, which is called beta-hydroxybutyrate.
Beta-hydroxybutyrate, or BHB for short. So to make this easier to remember, just think of BHB as a battery helper booster.
That's a great mnemonic, battery helper booster. And the evidence they found by just using this booster was staggering.
Yeah, giving BHB directly to the mice, or even just adding it to lab cultures, completely replicated the benefits of the full keto diet.
It massively enhanced the CRT cell's cytotoxicity, which is its killing power, and its proliferation.
And it wasn't just blood cancers, right? It even suppressed solid tumor models, like the HPC1 pancreatic cancer model.
Which is huge, because solid tumors are notoriously immunosuppressive and hard to treat.
Wait, let me clarify something, though. Is the BHB killing the cancer directly, or is it just making the T cells stronger?
That's a crucial distinction. While BHB might have some very mild direct effects on the tumor, its primary mechanism is vastly improving the T cell's metabolic fitness.
So it's basically buffing up the immune cells, not poisoning the tumor.
Exactly. But the catch is the BHB must be actively present in the blood circulation to work. You can't just prime them in a lab and hope for the best.
Right. It has to be a continuous supply. Okay, so we're at our 5-minute milestone. Time for a quick recap.
Sounds good.
Bullet 1, the ketogenic diet dramatically boosts CRR T cell therapy.
Bullet 2, the secret weapon inside that diet is the BHB ketone.
And bullet 3, BHB acts as a battery helper booster for immune cells.
Yeah, that sums it up perfectly.
So, a question for you listening. Think about your own energy levels. Have you ever noticed a difference in your mental clarity or your physical stamina when your body is burning different types of fuel, like fasting versus eating a ton of carbs?
That systemic shift you feel is driven by the exact same molecular pathways they are hijacking here to cure cancer.
Which is wild to think about. So let's go into the engine room, so to speak. How does BHB actually create energy inside the cell?
Let's break it down step by step. First, the BHB enters the T cell.
Okay, it's inside. Then what?
Once inside, it meets an enzyme called BDH1. That's beta-hydroxybutyrate dehydrogenase 1.
BDH1, got it.
This enzyme converts the BHB into acetoacetate and then very quickly into acetyl-CoA.
And acetyl-CoA is the magic key here, right? Because it completely bypasses normal glycolysis.
Exactly. It completely skips the glucose waiting line and jumps straight into the tricarboxylic acid cycle, or the TCA cycle.
The TCA cycle, which is basically the engine inside the mitochondria.
Right. And by feeding the TCA cycle directly, it drives oxidative phosphorylation, or OXPHOS.
And OXPHOS just churns out massive amounts of ATP, which is cellular energy, and NADH.
Massive amounts. And the paper had this brilliant way of proving this using isotope tracing.
Oh, the carbon-13 case. I love this part. They essentially tagged both glucose and BHB with heavy carbon-13 isotopes.
Right. They used mass spectrometry, so these heavy carbons act like microscopic GPS trackers. They could see exactly what fuel the cells were eating.
And what did they find? The T cells actually preferred incorporating the BHB carbons into their TCA cycle over the glucose.
Exactly. A good analogy is glucose is like your standard unleaded gas, right?
Yeah.
But BHB is this high-octane racing fuel.
And the mitochondria just eagerly devour that racing fuel.
Yeah, they do. And what's fascinating here is how the researchers definitively proved this dependency.
Right, because correlation isn't enough in biology. You have to break the machine.
Exactly. So they use CRISPR gene editing to literally delete that BDH1 enzyme from the CAR T cells.
So they removed the ability to digest a BHB entirely.
Yes. And without BDH1, the super boosting magic completely vanished. The cells were blind to the ketone.
Wow. Okay, time for a quick subtopic summary. First, BHB bypasses normal sugar metabolism entirely.
Second, the BDH1 enzyme is the key that unlocks the BHB, flooding the mitochondria with energy.
Perfectly said. So, question for you. If our cells have this backup high-performance engine for ketones, what other stress situations in human history might have trained our immune systems to run on this alternate fuel?
It's a profound evolutionary question. Famines, long winters, our immune systems had to stay lethal even when sugar was completely absent.
It's amazing, but we have to transition from the hardware, the fuel, to the software, because BHB doesn't just feed the cell.
No, it actually rewires its DNA expression. It's a profound signaling molecule.
Here's where it gets really interesting. How does a fuel change DNA?
So we're talking about epigenetic reprogramming. It's not changing the DNA sequence, but physically altering how the DNA is folded.
And they map this out using some pretty advanced tech, right? 8-TAC-sec and RNA-sec.
Yes. 8-TAC-sec basically looks at the physical structure of the chromatin, literally mapping what parts of the DNA are forced open or slammed shut.
And then RNA-sec looks at the actual transcriptomic output, like which genes are actively being printed into proteins.
Exactly. So the 8-TAC-sec data showed that BHB physically forces open what we call effector regions. It turns on fighter genes.
Fighter genes like FOXO1, right? I remember that from the data.
Yes, FOXO1 is a master regulator that promotes T-cell survival and memory. BHB cranks it wide open.
But it's a dual action thing, because while it opens fighter genes, it also slams shut suppressive regions.
Right. It aggressively condenses the DNA around exhaustion genes, physically turning them off-F.
And the main exhaustion gene they highlighted was TOX.
Exactly. TOX drives T cells into terminal exhaustion when they're surrounded by tumors. BHB literally silences that program.
And what about the tumor's defenses? Because tumors secrete these hostile molecules, right?
Yeah, tumors dump immunosuppressive molecules like prostaglandin E2 or PGE2 into the environment to sedate the T cells.
Right. But the BHB reprogramming helps with that too. The RNA-seq data showed it modulates an enzyme called HPGD.
Yes. HPGD is an enzyme that specifically degrades those exact immunosuppressive prostaglandins.
So BHB is literally giving the T cells a more resilient biochemical armor against the tumor's chemical weapons.
It's incredible. It degrades the tumor's defenses so the CRT cell can keep fighting.
Okay, we hit our 10-minute milestone. Time for a quick recap.
You want to take the first bullet?
Sure. Bullet 1, BHB is high-octane fuel for the TCA cycle.
Bullet 2, BHB is also a software update that physically opens and closes DNA.
And bullet 3, it actively turns on fighter genes like FOXO1 and turns off exhaustion genes like TOX.
Spot on.
So, here is a question for you to mull over. If a simple molecule derived from a diet can physically alter how DNA is folded and read in your immune cells, how might your daily dietary habits be subtly reprogramming your own immune system right now?
It definitely makes you look at your lunch a bit differently, doesn't it?
It really does. Okay, so curing mice is great, and petri dishes are cool, but what about actual human patients?
Yeah, that is the ultimate test for translational medicine. Does it work in humans?
So they looked at human clinical data, breaking it down into retrospective and prospective evidence.
Right. First, they did a retrospective analysis of blood serum from patients who had already received CART-19, specifically tisagenlecleucel.
Tisagenlecleucel. Try saying that 5 times fast.
I know, right? But basically, they found that patients with naturally higher ketone markers in their blood had better CAR-RT expansion and survival.
But that's just a correlation. It's retrospective. The really impressive part is the prospective study they did.
Oh, absolutely. They took completely healthy human volunteers, no genetic engineering, just normal people.
And they just gave them an oral BHB supplement to drink, just a ketone drink?
Yep. Then they drew their blood and used seahorse extracellular flux analysis to look at their T cells.
And the results were insane. Their peripheral T cells showed massive spikes in oxygen consumption.
Their oxygen consumption rate, or OCR, just skyrocketed. The mitochondrial membrane potentials surged.
And the overall ATP energy production just spiked. All from drinking a supplement.
Exactly. If we connect this to the bigger picture, it proves we don't necessarily need to force sick patients onto a miserable, strict keto diet.
Right. We can just use metabotherapy.
Metabotherapy. Just simply giving them the active BHB metabolite as a highly targeted supplement.
And this isn't just theory anymore, is it? It's already moving into the clinic.
Far from theory. It has already triggered an active phase I clinical trial.
That's the trial NCT 06610344, right?
Yes, specifically for human lymphoma patients who are actively receiving CRRT therapy. They're testing the safety and efficacy of BHB right now.
Which is just incredible speed from the lab bench to the clinic. Okay, let's do a quick subtopic summary here.
All right, well, first, BHB works on human T cells in real people, not just in mouse models.
Right. And second, metabotherapy, taking supplements, can effectively replace the need for strict, unbearable diets. So a question for you. Would you be more likely to completely upend your lifestyle with a strict medical keto diet, or simply take the active metabolite in a targeted dose?
The future of medicine might actually let you choose, which is a massive leap for patient quality of life.
It really is. Okay, let's bring everything together for our final review. We set out on a mission today to unpack this massive 2026 paper.
And we discovered exactly how the BHB ketone supercharges CRT cells.
Right. It acts as both a mitochondrial power-up, bypassing the normal sugar pathways, and an epigenetic software update.
It perfectly bridges the gap between basic nutritional metabolism and advanced synthetic immunotherapy.
And the relevance to you listening is huge. The food and the metabolites circulating in your body right now aren't just empty calories.
Not at all. They are highly active signaling molecules. They're communicating with your immune system on a microscopic genetic level every single day.
So what does this all mean?
Well, I want to leave everyone with a final thought to mull over. We've talked all about how introducing BHB can supercharge these engineered CRT cells to hunt down cancer.
Right, the synthetic super soldiers.
But think about your natural immune system. What is the everyday metabolic environment of our blood doing to our natural immune surveillance right now?
Oh, wow.
If a high glucose, metabolically stressed environment exhausts T cells, are we accidentally starving our internal defenders with the modern diet?
That is a fascinating way to look at it.
Could we one day have personalized immune metabolic profiles, like targeted metabotherapy to prevent disease from ever taking hold before it even starts?
To be basically optimizing our natural defenders before they ever have to see a tumor.
Exactly. The fuel we provide the system might be just as critical as the immune cells themselves.
That is incredibly profound. Thank you so much for joining us on this deep dive today.
It was a pleasure. Thank you.